Okay, welcome to the webinar. I'm Paul Hallett. I'm President-elect of the British Society of Soil Science, and I'm delighted to welcome you all to our 6th webinar of the year. There's 2 more to go before the end of the year. So before I welcome our presenters, I would like to introduce the British Society of Soil Science as the hosts of today's webinar. So we're an established international membership organization and charity, and we're committed to the study of soil in its widest aspects. So we bring together those who work in academia, and we also have a growing membership amongst practitioners who are implementing soil science in industry and those with a keen interest in soils. So we're hosting 8 webinars in 2023. There's 2 more to go, so please keep an eye out on our website for any further details on these. So before we begin, just some basic housekeeping. There's a lot of you on the call today, and all the microphones have been muted. What we'll be doing is taking questions at the end of both presentations, and we'll be monitoring these throughout the webinar. So please can you submit any questions you have by 12:50 to allow us to get through as many as we can. And although there is a raise your hand button, we're not going to be using that in the sessions today. Today's presentations have been awarded BASES and NROSO CBT points. If you're registered with either body, please contact us directly after the event. And finally, please be aware that we're recording today's presentation. So I want to first introduce our first presenter, and that's John Hollis. So John is a fellow of the British Society of Soil Science, and previously he worked as an independent consultant specialized in soil and water resources and the environmental fate of agrochemicals. So after 13 years of field soil surveying experience, he moved to the Soil Survey of England and Wales headquarters to assume national responsibility for soil classification and correlation. John was the NSRI's principal scientist, responsible for co-developing the Hydrology of Soil Types, or HOST system, which links soil physical properties to stream hydrological response to rainfall. He was also part of the group that developed the World Reference Base for Soil Resources, and he has authored or has been joint author of 25 peer-reviewed papers in scientific journals, 18 books or book chapters, including 7 soil survey publications with accompanying soil maps. And in particular, and we'll mention this later, John co-authored The Fragile Skin, Soil Landscapes of the UK, and that is what this webinar today is based on. So over to you, John.
Okay, thanks very much, Paul. Let me just show my screen, make sure it's okay. Can everybody see that? Is that okay? Right, well, before I start, I need to say a couple of thank yous. Firstly, to my co-author, Alan Lilly, whose knowledge and expertise on the soils of Scotland meant that we couldn't have produced The Fragile Skin in such a comprehensive overview as we do now. And then secondly, I'd like to thank Sarah Gary, the Executive Officer of BS Cubed, whose enthusiasm and encouragement enabled us to get the Fragile Skin published through the auspices of the BS Cubed and at such a reasonable price. I will miss her when she leaves this November, but I'd like to say a particular thanks to her. So, why have we called this talk The Fragile Skin, and why do we call the book The Fragile Skin? Well, there's increasing understanding that soils are under pressure from a number of factors these days. So much so that in 2018, DEFRA published a 25-year environmental plan, which recognised the need to improve soil health, there's that mention of soil health, by addressing factors in soil degradation. And that culminated in the current Sustainable Farming Incentive Scheme, which provides payments to farmers for actions for soils that are focused on improving soil health. There it is again. And you looking at structure, organic matter, biology. What do we know about UK soils? It's worth asking this question at this stage. Well, we actually know quite a bit. There's been over 90 years of research into characteristics and behaviour and survey of soils in the UK. And that's resulted in a lot of information that's now available online at various websites, one for England and Wales, one for Scotland and one for Northern Ireland. And there's been similar activity and information available in the Irish Republic, and we'll hear a little bit about that in the next talk. So we're now in a position to provide an overview of UK soil types. However, that's not really straightforward, as unfortunately there are different systems used in England and Wales, in Scotland and in Northern Ireland. And therefore, when we were looking at producing the fragile skin, we decided to use the World Reference Base. So this is an international soil classification system developed by a working group of the International Union of Soil Scientists and the FAO. And that intention was to act as an umbrella through which the various national soil classification schemes across the world can be correlated. And what we found is that we can produce a map like this for the whole of the UK, which shows 13 main soil types at the highest level in the WRB system. So the broad global level, this shows the main 13 types that occur in the UK. But to put that into a more global context, here's a picture of the FAO soil map of the world, where you can see various zones. This is a sort of classic overview relating to various soil zones across the world, and you can see areas that correspond to the steppes, the boreal forest region, the tropical rainforest regions, etc. And it's worth looking at where the UK fits into that pattern. Well, the UK is a small island. It occupies only 0.2% of the total land surface of the globe, and in a very natural, restricted latitude between 50 and 60 degrees north. But interestingly, it has it's got 22 of the main soil groups that occur. That's more than the 13 main ones shown on the map. And really that means it's a very diverse landscape in the UK for such a small area of the globe. Why is this UK soil landscape so diverse? We need to look at those factors that affected the development of those landscapes. And here we're looking at the classic Hans-Jenny factors of soil formation equation that says soil is a function of climate, organisms, relief, and parent material and time, and possibly some other factors. And this can be summarized in the fact that at any one point in the landscape, the local soil type is the product of the interaction of climate, organisms, and relief acting upon soil parent material over a period of time. And whilst I haven't got time to go into the details of the diversity of each of these factors across the UK, I'm just going to show two. All the others are detailed in the fragile skin. So if we start off with climate, There's two maps here, one showing the potential soil moisture deficit that builds up in the summer months, and the other showing the excess winter rainfall, that is the amount of rainfall that occurs in the late autumn, winter and spring, has to be disposed of through the soil. And what we can see is that when we're looking at the potential soil moisture deficit, it varies from less than 50 millimeters up to over 200 millimeters, whereas the excess winter rain ranges from less than 100 millimeters to over 2,000 millimeters. And for both those things, that's a very big range. So that's one thing. We look at the time factor, and this is something that you may not have seen before, but we've got four broad chronological zones. And these range from southern England, which was never glaciated, and where we've got landscape and soil evolution over about 2 million years period. And there may be remnant features in the soils that are as old as that. There's then an area in the Midlands of England, which is we've called the older glaciated landscapes, which have evolved over the last 4 warm cold cycles. And remnant features here may be up to 400,000 years old. Then the bulk of the UK is a recently glaciated landscape where we've got evolution only over the last 18,000 years. And in places only over the last 10,000 years. And then finally, we've got some anthropogenic landscapes where soil evolution has occurred only the last 400 years as a result of the installation of regional drainage schemes. So again, that range of time for evolution from 400 years to nearly 2 million years is enormous. And that's just 2 examples of the factors of soil formation. So what about the interaction of these soil forming factors within the landscape? Well, we can look at that using a sort of Venn diagram. So the blue circle represents the factors of climate, relief, acting on parent material over time, and that produces a set of morphochronological landscapes. The second one is where we've got relief and climate and parent material interacting to produce a set of hydrochemical systems. And finally, where we've got climate and organisms working together to produce a set of biological or ecosystems. And at the center of this is soil, a pedagogical example of the interconnectedness of all things. And I just want now briefly to look at what the interaction of those factors has done to produce a set of morphochronological landscapes in the UK. And here you won't be able to see the detail, but there are 22 of these landscapes, and each one represents a region with a common pedogenetic and geomorphological basis, within which the distribution of the different hydrochemical systems determines their inherent soil characteristics, and imposed on that, the distribution of different ecosystems determines the extent to which the inherent characteristics have been modified, particularly in relation to different topsoil humus forms. And again, I haven't got time to go into the details there, but chapters 5 to 8 of the Fragile Skin deal with each of these landscapes. But I'm just going to show 2 examples, a very quick overview from the north to the south of the UK. So starting in the south, we've got 2 examples from Scotland of the landscape and the soils associated with it. And here we're looking at very recent glacially sculpted mountain landscapes, one from the northwest of Scotland near the coastal areas, and the other in the Cairngorm Plateau. And you can see that there's a range of very skeletal bare soils to the start of soil evolution, where you've got some vegetation occurring, and through to a shallower podzol type soil, which is developed in the Cairngorm Plateaus. And to look at something entirely different, if we look at the ancient Thames and Solent terrace staircases, these are very old forms within the unglaciated landscape of the UK. And I think whilst you won't be able to take the details, you can see that there are some very complex features here in terms of the characteristics, the amount of soil evolution that's gone on, and that's reflected in the rather complicated long names using the World Reference Base. They represent this complex evolution. So why does this diversity matter? Well, soils support a range of functions that are beneficial. They can be said to have human capital. I'm sure you've come across that before. However, the ability of soils to support each of those functions varies according to their inherent characteristics. That means that the management of land with respect to any specific function depends on the limits imposed by those inherent characteristics. In other words, there is no magic bullet when we're looking at how to manage soils. Touch on that at the end. So just to look at how the interaction of the factors produce inherent characteristics that limit things. I'm just going to focus on, at this stage, on the carbon storage function by looking at the carbon profiles resulting from the interaction of various factors. First of all, we look at upland soils where you've got peat building up because of the climatic conditions, or podzolic soils where you've got a particular characteristic developed due to acid conditions. And here you can see the range of very high organic carbon contents down the profile from the peat soils, the fringe upland soils, the stagnopodzals on moorland, which have got a very high peaty top organic carbon peaty topsoil, and then shows the classic podzol profile of carbon, where you've got peaks in the immediate subsoil in the stagnopodzol, or in the lowland podzol a bit further down. This is a classic podzol characteristic. Moving on to the lowlands, here we've got a set of profiles that are conditioned by land use, water regime, and texture. And I've just highlighted those three differences. If you look at the water regime differences, so the blue groundwater clay arable and the brown earth free draining arable soils, you can see the distinction between the organic carbon contents of those two soils. Similarly, land use differences, the dark brown earths under woodland and the orange typical brown earths, free draining brown earths under arable. Again, quite a difference, although I would point out that the high organic carbon content under woodland, it doesn't extend anything like as deep as obviously the arable soil. And then finally, we've got some textural differences whereby the yellow brown sands and the orange brown earths are compared, and you can see there's a difference here in the organic carbon content of those two types, which is conditioned by texture. The fact that organic carbon can be strongly linked to clay content, and the differences between the two obviously limit the amount that can be stored. So what we can do is then use that understanding to develop a strengths and weaknesses table for the functional capacity of key soil types, the reference groups at the highest level in the WRB, showing in this case here the dark green colors show the highest potential for carbon storage, and the very pale colors the least. And we can do a similar thing in relation to water regulation and redistribution. This is based on the Hydrology of Soil Types system, which I can't go into details, not got time, but you can see again that we can do a similar exercise. And in the final chapter of the fragile skin, we do this along with an assessment. It's a sort of SWOT analysis. So these are the strengths and weaknesses. We then look at the opportunities and threats. And if you get the fragile skin, I would urge you to look at that last chapter, if nothing else. So to finish up, I just want to talk about soil diversity and soil health. Well, soil health currently has got a very high profile, but in soil science, we seem to be struggling a bit with exactly what the term means. However, if we focus on soil functions, then we can come up with some sort of definition which is along the lines of a healthy soil is one which can support a range of functions. And here I've highlighted a number of critical concepts here. Firstly, without significantly impairing any specific function. And secondly, up to a limit imposed by its local climate and inherent textural and hydrochemical characteristics. Now, that's a lot of words, but I'll try and cover it with just one example. If we look at regenerative farming, and in particular the COP22 4 per mil proposal, then we can say a healthy soil is one which can maintain its optimum organic carbon content without significantly impairing its potential to economically produce crops, reducing its microbial diversity, or increasing its nitrate leaching and N2O, NH4 or CO2 atmospheric emissions. And if we look at it like this, what this means is that firstly, we need to understand the optimal organic carbon content for specific soil types, which will probably be related to clay and calcium carbonate content. We then need to apply those agronomic management methods that minimize pesticide use and losses of N2O and NH4 as well as nitrate. And finally, to do that, we need to monitor or model at which threshold further increasing soil organic carbon may become unacceptable in terms of increases in CO2 emissions. And there is a limit there, I would stress. So finally, some take-home messages. Firstly, viewed at the global scale, UK soil types seem particularly diverse. Secondly, when we look at the management of soils, we need to understand it should be based on their multifunctionality, not just one of the functions, but all of them, in the light of the limitations imposed by their inherent characteristics. And again, I'll return to this, as I said it before, there is no magic bullet solution that we can apply to all soils. We need to tailor any remedies that we come up with to manage soils sustainably to the limitations that are imposed by their inherent characteristics. Thanks very much. I hope I haven't overrun too much.
All right, thank you very much, John. There's some great material in that I think I'm going to use for teaching as well. Just a reminder that we'll be taking questions for both speakers at the end of the session. So feel free to add any questions you have in the questions tab. And I would like to now introduce the next speaker, who is Dr. Rebecca Hall. So Rebecca Hall is a postdoctoral research fellow working on the Terra Soil project in collaboration with the Geological Survey of Ireland at Teagasc, the Agriculture and Food Development Authority. The Terra Soil project is using the archive of TELUS soil samples for chemical and spectral analysis to produce additional parameters on available phosphorus. Her research is mainly focused on biogeochemical cycling of phosphorus in soil and grasslands and its impacts on water quality. Her work has also looked at soil phosphorus testing and nutrient dynamics in small-scale experiments, looking at fertilizer, soil, root interactions to gain large-scale catchment modeling of soil structural degradation and erosion. And she started doing a lot of that work with me when she was undertaking her MSc in soil science at the University of Aberdeen. And she's since achieved her PhD in biological sciences from Queen's University Belfast. So over to you, Rebecca.
Great. Share my screen. Can you see that there?
Yep, it's all good.
Brilliant. So yeah, I'm just going to talk through mapping and predicting soil health indicators in Irish soils, but just to start by giving a little bit of background. So I'm working at Teagasc, the Agriculture and Food Development Authority. Sorry, I've got a lot of things on the screen here. Based down here in Wexford. We're working a Crop and Environment Land Use Programme in Johnstown Castle, which is a lovely place to work. So I'm not a geologist, so I'll just give a brief overview, linking this to the previous talk. So Ireland had 3 successive glaciations, which gave it a really heterogeneous geological landscape. In Ireland, the Midland areas are quite flat, but there's a lot of mountains too. On the western coast of Ireland, there's loads of rainfall. There's a lot of rainfall in Ireland, but even more there, which is give way for a lot of peatlands. So on the west coast and through the Midlands, we're pretty much covered by peat soils and surrounding them organic mineral soils. Then we'll go through to the east coast and we'll have a lot of mineral soils. So there's a good cover of great soil groups, mainly brown earths and some glaze, but there is a great cover of them. So the Irish Soil Information System started back in 2008. I think it was the latest version of it. So it had 58 soil associations, excluding the Abrian areas, alluvium soils, peats and rocks and marsh. So what they done was they had a soil core and auger for 11,000 locations, but they also had 225 soil pits where they excavated and done really in detail soil sampling. So there's a really good archive of soil samples there to use. And then going down a more local scale, there's 150 soil series detected there. So the project I've been working with is the Geological Survey Ireland, the Teagasc Geological Survey. So it started up in Northern Ireland, but we don't really work with the data. We've just used from the borders, the West and the Midlands. So we collected the Geological Survey Ireland collected the A samples and the subsoil samples on a 2x2 kilometre grid and a bit smaller resolution in Dublin and Galway. So that's collected for ICP agroregia analysis, XRF, pH and organic matter. So when I started my postdoc back in 2020, we just had all the samples came through and we analysed the A samples for nutrient analysis and the top and subsoil samples for carbon and particle size. So the aim of this was to make agronomic indicators to see what regional trends are going on, because like John said before, there's not a one fits all answer for soil health, and because we've got such a heterogeneous landscape, we thought if we analyse this spatially, it might give more idea to have better agronomic practices. So in Ireland, the biggest problem we have is phosphorus. So what happens is a lot of phosphorus is put onto the land, but the majority of soil samples that are collected annually by Teagasc is low in phosphorus. Even though there's a lot of runoff coming from the soils and a lot of fertiliser going on, that phosphorus just is getting locked up in the soil. So what we done was we took all of the samples and we took the nutrient samples just for the topsoil. So the problem with trying to understand this phosphorus problem and looking at the geospatial characteristics of it is it's really all down to phosphorus additions like manures, fertilizer. You've got to take into account plant uptake, crop removal, erosion, runoff, and then any sorption dynamics, which is driven by the geology. So there's a lot of work went into mapping phosphorus, but it's just very complicated and it's not just straightforward as wondering what's going on there. So now I'm talking about phosphorus. I'm talking about ICP, which is the total fraction, pretty much. Muriatic phosphorus, which is ammonium fluoride extraction, so it extracts aluminium aluminium phosphate, calcium phosphate, and iron phosphate. And then the plant available fraction. So in Ireland, we use Morgan's P. Over in the UK, it's Alsam P. So there's slightly different extractions. But here it's calibrated against crop yield to give an index system of how much fertilizer you should use. So what we found was when you just take all of the data and you map it, there's no coherent distribution, and it doesn't seem to be linked to underlying geology. It doesn't really match the patterns of peatland or even any stock and rate. So the majority of the landscape here is agriculture with grassland agriculture. So you would have thought it would kind of match cattle, but it's not, because depending on when you take the sample, it could be different one day to the next, which is why it's so tough to model phosphorus. So one way that we looked at it, because we're publishing these interpreted maps. So if you're going to put them out there, you need a kind of a warning sign with it. So in the top left-hand corner, what we have is a variogram map. So I always say if they look like a QR code, then it's not going to be coherent in spatial distribution. But for example, aluminium, plant available aluminium is. So you can see the difference in that mapped variogram there. So the geological distribution in Ireland has really high aluminium soils coming from the parent material, especially in the regions we're looking at. So we found in a colleague in a previous study found that mealic aluminium is a direct indicator of soil sorption behaviour. So there's a cut-off where above that, no matter how much phosphorus you apply to the soil, it's going to take absolutely loads of it to not just completely absorb. Now, on the other hand, below this category, you're going to have soils that don't bind phosphorus very well at all. So I had a look at soil index in comparison to mealic. So you've got low, very low. Moderate would be where you don't have too much runoff and you sustain crop yields, and high is there's no benefit to having any fertilizer on at all. And so the red line here is that cutoff value from the mealic aluminium. And so just a basic ANOVA found that the aluminium does have an effect on the phosphorus, and the majority of samples were low in this aluminium category. So that's what we mapped. So on the left here, you've got your high P fixing soils, and on your right, you have your low P storage soils. I'll not really go too much into the low P storage because we're hoping that the further down we'll go with the survey, the better it'll be to show that kind of behaviour. But in this part of Ireland, we have loads of these high aluminium, low phosphorus soils. So this whole region is where you're putting loads of fertilizer on, and it's not always having too much of a difference on the yields. So what we looked into a bit further was the total phosphorus and potentially labile phosphorus. So the top map there is the total phosphorus, and the bottom map is the kind of probably readily available phosphorus depending on management. So in these areas, you do have a lot of phosphorus. It's just with the phosphorus test we're using, it's not necessarily plant available. So just as a kind of a validation proxy, we looked at the pollution potential maps from the Environmental Protection Agency and found in these two regions where you've got the most pollution from agriculture, where in the first map of phosphorus I showed you there, the pressure of agriculture on waters, lakes and rivers, they're mainly surrounding in these areas, we've actually got really high amounts of phosphorus. It's just the soil test that we're using isn't necessarily picking that up. But luckily, we've done another study in my postdoc, which looked at converting mealic to Morgan's. So you can use random forest machine learning algorithm to use aluminium, calcium, iron, and phosphorus and pH, all from mealics, and you can predict out a soil index in Morgan's P. So 90% of the samples predicted the correct soil index or even concentration within 1 milligram per litre. And so the bottom graph here is showing the external validation. So we had, I think it was about 500 samples from other published studies that have used the same data. And we found that it did predict really well, unless it was outside of the survey area. But because Ireland is such a heterogeneous landscape, it's hard to really always say exactly what's going to be going on in those soils. So again, back to the point with trying to develop soil health indicators and looking for soil health monitoring law that's coming from the EU, it's not necessarily just a one rule, a one soil function. There's going to be a lot of data going to understanding how you can actually have what is a healthy soil. So just finally, just quickly, we're also developing spectral libraries in Teagasc. So I'm part of some of the studies, but mainly with my colleagues are looking at spectroscopy for different soil attribute and soil health indicators. So I'll just play this video here. So spectroscopy just uses a light source and it goes through the computer, and you calibrate that against an original data set. So we're using the TerraSoil data set. So we've managed to successfully predict and publish using spectroscopy for particle size and texture to predict pH, cation exchange capacity, different forms of carbon and percent organic matter. PhD student Longland's still working on predicting carbon stocks, because obviously you've got to build up the carbon before you can do that. And then, but most important, this absolutely doesn't work for nutrient status. You have to do the prolonged wet chemistry that I was just talking about there. So the soil health indicators that you can predict and are published are line requirement. A PhD student, Sifan, is working on using it to predict the absorption capacity that aluminium thresholds I was talking about, and Longland's working on the carbon sequestration. And on top of that, we'll also have an XRF. So XRF and MIR combined can help predict some of these things. So for example, the carbon work that we're involved with the PhD student Longland, this is the original data from the Teagasc geochemical survey. So what we're doing on top of that, we'll look at soil organic carbon, bulk density, and soil total carbon. We run that through the reference data, so we'll use 1,000 samples, use 70% for the calibration and 30% for the validation. Then this is the spectral data that you get from all of the 10,000 samples. We use chemometric modeling and machine learning algorithms to convert this into a value. And so far, again, published, we've got carbon and percent organic matter. And we're working on carbon saturation potential, think about carbon sequestration. But Longland's just published a paper recently predicting bulk density class. So as Paul's a bulk density soil physicist, it's just a caveat here. It's mainly just for carbon stock calculations or comparing it to pedo transfer functions. It's not really for detailed, compaction characteristics, but compared to pedo transfer functions, it works really well, the model, if you want to give that paper a read. And then finally, what we're doing to develop more of these soil health indicators is using benchtop to calibrate handheld. So potentially we could take this into the field. So any further surveys you can get that bit more information at a quicker level. So yeah, just to reiterate, there's a lot of work in Ireland, especially with the soil health law coming into place and to be net zero by 2050. So using this geochemical survey to develop these indicators is very helpful when you've got heterogeneous landscapes. So this was a huge team effort, all of the work that we've done. So thank you to the team, and thank you for inviting us to this presentation.
All right, thank you very much, Rebecca. That was really, really fascinating. You've done a huge amount of work as well. We're now going to open up to the question and answer section of the webinar. So I'm pleased to welcome James Dower. So James is a pro at doing this, and he's one of our early career committee members. And he's been monitoring the questions while you've been watching the talks, and he has assembled them just in order to start putting them to them. So just a reminder, if you still want to submit a question, you still can do by 12:50, and this will give us a chance to get through as many as possible. So I'll pass over to James now.
Good afternoon, everyone. Yeah, as Paul said, please keep popping your questions in the chat. I've got one here, and I'll start with you, John. Do you think that the apparent diversity of UK soils has anything to do with the comprehensive surveying that's been done? Or in other words, if other countries looked very closely, would they also have a range as wide as we do?
That's a very good question, which probably sums up in the more you look, the more complicated it gets. But there are many other countries that have as comprehensive a set of information available about their soils as we do in the UK and Ireland. So I'm sure there is an element of what you're saying. The more you look, you know, the more complicated it gets, and certainly that relates to scale. It's still quite complicated at small scales, but I think in general, because of some of the rather unique characteristics of the UK, it is more diverse than you would expect for such a small area. I think that's all I'm saying.
Cool, thank you. Rebecca, this question for you. What kind of methods have you used for P mapping?
Oh, we've used a lot. We've spent ages trying to do it because a lot doesn't work. So we started off by using all the texture data, pH, loss and ignition, everything, and threw it all into a random forest model and tried to do some more like in-depth modeling, but it just didn't show anything. So we then tried to do the Kriggen, but again, the variogram map showed that there wasn't much spatial coherence. So that's why we'll kind of keep them on the whole time. So yeah, where we've got the aluminium and the phosphorus together, the two separate maps, we used IDW to then make a contour, and then that's the data that we have there. And then when we put the other data within that, that's Kriggen again.
Cool, thank you. Back to John. What do microbes and rhizophagy play in evolution of these soils?
Well, sorry. So this is part of the biological, the ecosystems component, and I'm sure you don't need me to tell you that. So microbes and do you say macrophages or microphages?
I said rhizophagy.
Rhizophages, sorry. So the rhizophages and the microbes play this role in fixing carbon within the soil profile, but it very much depends on how they're distributed down the profile, and they tend to be, well, obviously the rhizophages are associated with rooting. And if you look at rooting in general, what you find is that most roots, possibly 95% of them, are actually focused in the top 30 centimeters. So that's inevitably where most of that activity occurs, and they're responsible for fixing, for developing these carbon, clay, and possibly calcium carbonate, where that's present, complexes that stabilize organic carbon, I think.
Thank you. Rebecca, you spoke a little bit about spectroscopy. Do you think we can use spectroscopy to measure soil health? And what soil health metrics are currently available, maybe in Ireland or elsewhere that you know of? So
yeah, I think it can be used. I don't think it can't possibly replace the original wet chemistry methods, because you're going to have to continue to validate and build up the models. But as long as you've got the good spectral models to do the predictions, I do think it'll play a part, especially with, there's going to be a lot more going in agricultural monitoring soon. So it's going to really help the process. So at Teagasc currently we'll only ever work with anything that's been published. So the technologist, Felipe Bacchion de Santana, he does all of the modelling. So it's mainly just the carbon work that we're working with so far. But it's not replacing the wet chemistry.
Thank you. So some of the handheld ones you spoke about, how much calibration do they need? Do you think they could ever be like commercially widespread?
I don't know yet. I think you've got to be careful with them, because again, you can't completely replace the wet chemistry, and it's just the beginning of the research that we're doing here. And you're going to have problems with moisture content as well. So there's a lot of things that play into that. So I do think they've got a lot of potential, but I'm not entirely sure just yet until we've got the data.
Cool, thank you. John, there's a question here for you asking about whether you think England and Wales should also do P sorption work. Apparently Scotland have it as a basis for P recommendations, but we seem to stick to Olsen.
Yeah, I have to say I don't know enough about that. There are always arguments about which analytical method to use, and I think you need to talk to somebody who's a P expert rather than me on this. I'm sorry, it's not a very good answer, but I don't think so. The problem is that once you build up a certain amount of data about, on soils, historically, then the tendency is not to change your analytical methods, because you will get different answers. So that may be a factor. But as I say, I don't know enough about the details of phosphate chemistry, and certainly the analytical techniques related to. Sorry.
No worries. Rebecca, you mentioned a bit about the differences at Morgan's and Olsen's and this sort of thing. And this question says Ireland are developing information around P sorption. Do you have any comments on, whether England should look closer at sorption?
I don't know too much about phosphorus in English soils. I've done my PhD in Northern Irish soils, so I could answer maybe on that a little bit, where I think, mainly each soil test is developed for a different type of soil. So if you've got lots of different types of soils, there's less chance that it's going to be universal. So depending on your soil type, there's going to be a test that better reflects it. So you could introduce these conversion equations, or you could just have a look at where, anywhere that's got a lot of runoff or a lot of eutrophication, maybe that's a place to start. But I don't particularly know for English soils.
No, that's great, thank you. John, there's someone here that's curious about US soil taxonomy. Are they used in the UK at all, or do analogous schemes exist?
So the US soil taxonomy is widely used. It tends not to be used, but quite early in the evolution of the soil survey of England and Wales, we looked at how the soil classification we used fits in with soil taxonomy. So there is some work to do with that early on. My preference, soil taxonomy is incredibly complex, and it's got about 6 levels in a hierarchical system, and that can cause complications in the type of taxonomic separations that you use. The more of levels that you've got in a hierarchical system, the more likely you are to make some unnatural splits. And personally, I hate soil taxonomy. It's very intimidating. I don't think it's that helpful in explaining soil patterns in this country. I much prefer the world reference base, which has only got 2 levels. Basically, this top level I talked about, which relates to the broad global distribution of soil types, and then a whole set of at the next level down, the second level, you can add a whole set of qualifiers to it, which tells you a lot about the different ancillary processes that are going on in the soil and have created specific types of characteristics within that broad soil type. And that's why it's useful for correlating national systems, which can get very complicated.
Cool, thank you. Rebecca, there's a question here that says, if I sample for agricultural purposes only, I'll ignore, or if I sample for agricultural purposes, I'll ignore hotspots like feeding areas. But to look at environmental risks, those are the very areas we should be sampling. What approach to that kind of variability or sampling strategy did you take?
So the telesurvey stayed away from any contaminants. So if there was a feed in place or you know, a lot of manure on the ground, they wouldn't have took anything from there. So for the study we've worked on, it hasn't avoided those hotspots, although, you know, there would be times where you couldn't absolutely guarantee that you've avoided the contamination area. So I think that's why it's quite tough to model phosphorus. Like, what do you pick? You know, there's papers with in-field variability of just phosphorus. So I suppose it depends what your outcome, what you want to be modelling. So we wanted to have a look at a regional geospatial view, looking at different soil tests, different phosphorus pools. So that's why we went with this, using the Terra soil data. But if you were wanting to look at runoff, I would absolutely have a more targeted sampling approach for that.
Cool. There's another sorption question, and Rebecca, I'll ask you this. Can we have high levels of sorption, but also have runoff and eutrophication? Yeah, what do you think?
So, yeah, absolutely. So if you've got loads, so with the work we had where we had loads of phosphorus sorption, they are particularly bad in leaching and runoff, because there's a lot of it there. If you're ever going to have a lot of phosphorus there, it's going to be a risk, because it's not just the runoff, it's also the erosion. So there was some areas of really high aluminium with quite low phosphorus, but they were mainly outliers. We kept them in, but yeah, so if there's loads of phosphorus there, there's some way it will get into the water. So I suppose it's more about understanding what's happening and how that phosphorus is behaving on a smaller scale.
Thank you. John, would you imagine there's a significant shift in soil parameters from interventions, I should say, such as acidification of slurries? Have you factored anthropogenic factors into sampling and analysis? Not that you did any when you wrote the fragile skin, but yeah, what do you think on that?
Yeah, I'm not quite sure about that question, because one thing I would point out about England and Wales, maybe not so much in Scotland, but there isn't really any systematic sampling going on anymore. So what we tried to do when we looked at the national soil inventory, which was sampling at 5-kilometre grid intersects, this was to avoid the bias that takes out kind of areas with a strong anthropogenic effect input from ordinary soil survey, if you like, free soil survey. So the National Soil Inventory should have included those anthropogenic factors, and that should have been reflected in the land use definition that was added to each inspection point. I think that's about all I can say, unless there's something else that I've forgotten about and not answered.
No, that's great, thank you. A question's come in that I think it would be interesting to hear both of your answers on. You both spoke about a bit about soil carbon, and then Rebecca, you spoke about like fertilizer use, which obviously has a carbon footprint. So the question is, what do speakers think of the chance for the UK and Ireland to reach net zero agriculture? Or by 2050, you know, England has a 2040 deadline the NFU has published. So what do you think? Rebecca, should we start with you?
I'm not an expert on soil carbon. I'll not speak as confidently about it as phosphorus, but I think as long as you don't use offsets to cover up other practices, maybe, I think it's good to aim for something. I'm not sure. I'm not sure if how confident I am. Yeah.
What do you think, John?
Okay, well, if you'd have asked this question a couple of years ago, I would have said no chance. But there's been a sea change, I think, in attitudes towards this, and even among farmers. I went to help out BSQ at the latest groundswell event, and I was absolutely stunned by the number of farmers who were coming up and talking about how to maximise their soil carbon and improve soil health, etc., etc. I still think it will be very difficult for two reasons. One is a lot of people are jumping on the bandwagon and providing this magic bullet solution to increasing soil carbon. That's the first thing. And the second thing is, I think it will need financial incentives, and those financial incentives should be targeted at those soils where you stand the best chance of maximising the organic carbon content without increasing carbon dioxide contents. And I just wonder whether the country, and particularly its governments, have got the guts to, possibly the finance to do that. So I'm much more optimistic now than I was, because there is, as I said, a real change, but it still requires a lot of work.
Thank you very much. Thank you to the audience for all your thoughtful questions, and thank you, John and Rebecca, for your insightful answers. I'll hand back to Paul now.
Okay, thank you very much, James. And on behalf of the British Society of Soil Science, I'd also like to express our thanks to John Hollis and Rebecca Hall for coming along to present today. Those were excellent presentations, and they really fit into the framework of the fragile skin. So if you'd like to find out more or purchase a copy of the book, The Fragile Skin, Soil Landscapes of the UK, which was co-authored by John, you can do so via the British Society of Soil Science website shop, the link to which will be in a follow-up email. So thank you all for attending. You will find a quick feedback survey when you leave the webinar, which we hope you will take the time to complete. The recording of this video will also be available after the event on our YouTube channel. We want to bring to your attention our upcoming joint annual conference that we're hosting with the Soil Science Society of Ireland from Monday the 4th to Tuesday the 5th of December in Belfast. And this is going to be an excellent event. Registration's still open, and you can still submit an abstract for a poster. Right after that, we're hosting the Early Careers Conference on Wednesday the 6th and Thursday the 7th of December. And this is for our early career members. And this is also being held in Belfast. And again, registration is open, and you can still submit abstracts for posters for that meeting as well. So the next webinar is scheduled to be on Wednesday, the 4th of October. So keep an eye out on our website for how to register for this event. So hope to see you at future events. And in the meantime, thank you again and goodbye.